Greetings, ladies and gentlemen, and it's my pleasure to present our study, which is titled The Effect of Pretreatments on Microbiological Quality of Dry Tomatoes During Storage. Tomato is one of the key vegetables that is grown in Kenya and ranks second in terms of economic importance among all the other vegetables. However, due to its perishability, it has high post-harvest losses, which is considered to be as high as 65%. Such high post-harvest losses leads to considerable loss of income, not only for the farmers, but for virtually all the players within the value chain. One technology that can be utilized in preventing post-harvest losses is drying. This is because drying has the ability of lowering the water content of the product, and this is able to slow down or virtually eliminate any microbiological activities that contribute to spoilage. In drying systems, there's usually a number of pretreatments that are carried out to be able to preserve the vegetable. However, there is limited information on how these pretreatments are able to affect the microbial quality of dried tomato. Therefore, in this study, the objective was to determine the effect of specific pretreatments and drying systems on the microbial quality of tomato during storage. The Rio Grande tomato variety was grown under greenhouse conditions and was then harvested at mature red ripe stage. And fruits of uniform color, size, and shape were selected and then subjected to the various pretreatments, which involved spraying with 0.5% sodium metabisulfite, 0.5% calcium chloride, and also the control was distilled water. The drying was carried out under 4 conditions, that is oven at 70 degrees Celsius to a moisture content of 13%, vacuum oven at 70 degrees to a moisture content of 13%, solar drying, and also freeze drying was then carried out at minus 47 degrees Celsius at 0.055 millibars. After the drying, the samples were then divided into specific groups, and then they were subjected to microbial analysis monthly for up to 6 months. We realized that the pretreatments were able to affect the visual coloration of the dried products relative to the control. In addition, we realized that the specific pretreatments were able to suppress the total bacterial count, whereby the sodium metabisulfite suppressed bacterial growth compared to calcium chloride. However, even up to 6 months, the microbial count was still within the limits set out for dried tomato by the Kenya Bureau of Standards. For fungal count, similarly, we find that the sodium metabisulfite was able to suppress the fungal growth throughout the 6 months. And similarly, even after 6 months, the fungal count was still within the allowable limits set by the Kenya Bureau of Standards. In conclusion, we can say that bacterial and fungi were present in all the dried samples, but in all the instances, they were still within the limits. And the low microbial counts were indicate that the drying methods or the handling conditions were able to inhibit the growth. Sodium metabisulfite significantly reduced both the bacterial and fungal count compared to calcium chloride. But of course, we realize that calcium chloride can also be used. For more information, you can reach me through my email as indicated. Thank you for listening.